Okay, so we're gonna start our presentation, guys. Tandis, will you be sharing your screen?
Yeah, I'm gonna share my screen. Okay, you guys can see this, yeah?
Yes, we
Yeah.
can.
Okay. Um, okay. So you know the topic. Casey explained that it's gonna be about neurophysiology. I'll talk about the neurophys part, and I'll start with resting membrane potential. I think you all know what resting membrane potential is. It's like the potential, the difference between the inner part of the cell and the outer part of the cell in resting situation. So resting means there's no stimulus, nothing is stimulating the nerve, and this resting membrane potential, it is maintained by two main channels. One is the sodium potassium ATPase, and one is the leaky channels, which the sodium leaking channels and the potassium leaky channels. And there's a way to calculate this like resting membrane potential, and it's the equilibrium potential, which we use this formula over here. It's better to know this because they might ask. It's really basic. Just memorize the formula. You don't need to use it. Just know like what is what. And then
They ask questions like about like what's proportional to like an increase, decrease, right? I remember.
uh no, it's not in this one. That's like later for CVS and stuff. Because the formulas here, they're a bit complicated to ask that, so they won't ask. Just know like the formulas, like and what's there, and like basically what's related to equilibrium potential and what's related to Goldman equation. Like know what they are, but you don't need to know the relations and stuff. And after that, we have the Goldman equation, which it explains the magnitude of membrane potential upon the distribution of sodium, potassium, and chloride. So basically, you're calculating um, the concentration and how they are related and how they are maintaining the membrane potential. And oh, okay. This one is important. Like they ask a lot of questions about this. For sure, know the difference between non-conductive membrane and conductive membrane, and like all of this stuff, like the reactivity to stimuli. What is the difference? So like non-conductive is chemical, and the other one is electrical. The other one, like the conductive one, is only depolarization, but non-conductive is depolarization and hyperpolarization. I'm not gonna go through all of them because we don't have time. But know everything in this. Table, and sorry like it's so annotated and like untidy, but this was the only picture I had. So study this really well. And then okay, so the main stuff that I'm going to be talking about is how action potential is caused and how it's spreading like throughout the nerve. uh First of all, we said that we have a resting membrane potential. It's like something stable. It won't change unless there's a stimulus. When the stimulus happens, you will start, no sorry, the nerve will start um having like changes, like the membrane will have changes in the permeability. So first what happens is the voltage rises, and sodium channels, they open. The slow sodium channels, they will open and they will cause depolarization. After it reaches a certain threshold, which is 15 millivolts, the which we call it firing level, the fast voltage gated channels open, and we will have a like high raise and a fast raise until it will reach positive 35 or like positive 30. After that, when it reached that peak, like it will start repolarizing. How does that happen? By the help of the sodium, by the help of the potassium channels. The And we know that the permeability to potassium, it's much more than sodium, so repolarization, it happens like much faster than depolarization. And that's why it, like every time when this happens, there will, the membrane, it will undergo hyperpolarization, and for it to get back like the ion balance, it needs the sodium potassium ATPase. So the sodium potassium ATPase, it will bring it back. It will balance the ions and it will bring it back to the negative 70 millivolts, which is the main resting potential. It's really simple. You just need to know this graph and like which channels open at what time. And then, okay. So here in this slide, you will see changes in excitability during action potential. What causes change to this like whole process happening? One is local response. It's basically when we have a stimulus, the stimulus should have a certain intensity to cause action potential. If not, it won't cause action potential. So this threshold, it's important. If it won't reach that threshold, we call it sub threshold, and it won't cause an action potential, and basically nothing will happen. And then after that, here we have the absolute refractory period and the relative refractory period. In this period, first of all, in the first one, the absolute refractory period, which is basically from the beginning, from where the firing point starts, like you'll see it in this graph, until 1/3 of repolarization. You can see number 4 is still that part. That's the absolute refractory period. Whatever stimulus like comes again, it won't cause an action potential. Like nothing else would happen at that point. But in relative refractory period, a strong stimuli can cause excitation. So know the difference between these two. It's important, even like later for CNS and stuff. It's really important to know this. And the after potential periods, they aren't that important. It's just like know what they are. Negative after potential is the threshold of excitability decreasing, but positive it's when it's increasing. And it can be due to different reasons. You don't need to know that. Just know like what they are. And now, okay, stimulus. I mentioned the quantity, that like it should have a certain quantity, a certain intensity to cause action potential. And there are three other things that is really important when it comes to the stimuli. One is the quality, whether it is chemical, electrical, or whatever. And it also depends on the receptor, so like whether it's affecting like proprioceptors or stuff like that these all affect the quality. It's written in more details in your slides, like in the PDF you have. And then after that, after like except for quality and quantity, the time of duration is also really important. So the stimulus has to be there for a certain duration to cause action potential. If not, it won't cause action potential. You have a graph for it, which is this graph. It has a hard name. I'm not going to even try to pronounce it. And these are the stuff you need to know about it. First of all, I'll talk about Rio base. So this graph you see is basically telling the relation between the stimulus strength and the duration. Rio base is the minimum voltage that you need to cause an action potential. Chronaxia is the minimal time you need to cause an action potential, and it's basically twice as much as the real base. You know, like, so you have a real base is the minimum, but you need twice as much as that to cause a peak and cause an action potential. I hope you guys understood that. It's a bit complicating, but just like know what they are and like how they will affect the action potential. That's it. And like try to understand the graph. It would be fine. And the last thing that will affect stimulus is the rate of intensity change. It can change, like the intensity can change, and the action potential can be like more or less, or like it can stop and all of this stuff. Okay, another important topic here is the propagation of action potential, that's how it happens. So when a certain point in the nerve, like the stimuli comes to a certain point in the nerve, how does this like go along the axon? This will happen because of the change in permeability. So like in that point, the stimulus hits a point on the nerve, there will be change in the permeability, and then these ions, they will move throughout the axon, like on the inside, and they will change their permeability while they're moving. from the inside, and this will cause the action potential to move forward and move down the axon. And at the end of the axon, like in the axon hillock, there will be like formation of vesicles and neurotransmitters, which Katie's gonna be explaining how like the synapse works and how that process is. But here the main focus is on how it's going along the axon, and it is basically because of change in the permeability and how the ion concentration on the inside and the outside of the cell is different. This is like the general overview. We have a type of conduction which is the fastest and is 50 times faster than normal conduction, which is called saltatory conduction. If I'm wrong, sorry, but it jumps from one node to another node of Ranvier, and this is because like the myelin sheet, we know that it can't like basically transfer the ions, like it just makes it jump. It makes the stimulus just jump from one point to another, and it will make it faster. This is usually in muscle, like in when it comes to like muscle innervation, it's really important, because it would be, well, it's much faster, so we need this one, so study this really well. And then, okay, the type of conductions, we have orthodromic and antidromic and also local. The difference between these is like the direction of how it's going. So we know that axons, they have the ability to conduct in both direction. In antidromic, it's like the opposite direction, but orthodromic is as the same direction, like it's going downwards, but antidromic is going upwards. And that's all. Oh, and you have a few slides about how you measure action potential. There are a lot of details about it and like how the whole system works and stuff. But the main mechanisms, like the main techniques they have, is either biphasic action potential or monophasic. In biphasic, they will be basically measuring the action potential change in two parts of the nerve and two parts of the axon. But in monophasic, they're doing this in one part. So, like in biphasic, you'll be comparing two action potentials happening in two different parts of the nerve. But in monophasic, you'll just be talking about like one part of it. That's the main difference, and that's all you need to know. And yeah. Now Katie will continue talking about synapses.
Yep. And for this monophasic, biphasic as well, it's really important because the way you measure like the amplitude of the action potential, they had quite a few questions actually on that. So, but guys, all this concept, it sounds really complicated, but for me, once I watched Ninja Nerd, it was all pretty easy to understand. Give me a second, let me share my screen. Can you guys see? Can you guys see my screen?
Yeah, we can see your screen.
Okay, sorry, I can't see you. Okay, so I'm going to be talking about synapses. You guys know what a synapse is? Can anyone unmute and tell me what a synapse is? Anyone? Does
Do you
nobody
want
know
me
what
to tell you what a synapse is? Apparently, like no one is answering, so I'll tell you what a synapse is.
Yes,
It's
please tell us.
basically where the transmission of the stimuli happens. Like a
Yes.
really general explanation.
Yeah, it is where the stimuli is transferred from one neuron to another. So we have, like, if you can see in these pictures, can you guys see my arrow, my cursor? Tandis, can you please answer?
Yes, yes, we can see.
Yes.
Okay, perfect. Thank you, guys. So you have one neuron, and the stimuli that's present in one neuron, it transfers. What Tandis was saying about the non-conductive and conductive, the soma part, the body of the neuron is the non-conductive, while the axon is conductive. So the stimuli is transferred from one neuron and it attaches to the soma of the other neuron, but it doesn't always have to be the soma. So that's where we can divide synapses morphologically. And we also have another way of dividing synapses according to its character of transmission. So you guys did this in anatomy or histo as well, I think, the morphological division. It's where it doesn't just have to be from one neuron's axon going to another neuron's dendrite, but it can go from one neuron's axon to the other neuron's body. So it doesn't have to attach to the dendrite. And there can also be from one axon to another axon. And there can also be from one dendrite to another dendrite. Now, there's obviously like reasons why this kind of division is present. Like it could be to maybe inhibit some sort of stimuli from passing on. It could be to excite. And you don't need to know, like, I don't think you need to know the reasons why, but just know that there's these different classifications. Now, talking about the transmission character, we did not know about this before. Like we haven't studied the different transmission characters before because we've only learned about the chemical synapse, which you guys I'm sure are all aware of. Even Tandis addressed it with the transmission of neurotransmitters. But in this case, when we talk about transmission character, we have three different types. So we have an electrical synapse, we have a chemical synapse, and a mixed synapse. An electrical synapse is a synapse where you have this connection between the presynaptic and the postsynaptic, but you don't have a neurotransmitter that passes through it. It's instead an electrical synapse. So you don't need a chemical to move through. It's just the stimuli passes on from one neuron to another. And how this is able to take place is because the synaptic cleft, that means the space between the presynaptic membrane and the postsynaptic membrane, is very, very small. So you have easy leakage of ions. Now, a synapse, how it works is because of ions, the presence of ions like calcium, it causes like some, not calcium, sorry, sodium ions. That's what causes the stimuli to pass along. And because this influx of sodium is what is the stimuli pretty much, and the influx of potassium ion, the outflux, I think that's what it's called. The outflux of potassium ion removes this stimulus. So it's just these movement of ions that actually gives you a stimuli overall. The whole point of having a neurotransmitter is just moving from one neuron to another. But if you have a very small space in between, you don't need this neurotransmitter. So that's the whole point of an electrical synapse. It has a synaptic delay of 0.05 milliseconds, which is a very short delay. We don't really normally have electrical synapses in the human body. I think we maximally see it in some cells in the brain and even in the heart. I think in the gap junctions of cardiomyocytes, you have electrical synapses. Then talking about the chemical synapse. So in chemical synapses, our synaptic cleft is way larger. We can't just pass ions to transmit a stimuli from the presynaptic neuron to the postsynaptic. So we need the presence of a neurotransmitter. I already told you guys there's the presynaptic part, which is the first neuron's axon has a presynaptic part. There's some contractile protein that's present that helps in the movement of a vesicle towards the end. I'm going to come to that. And it's activated by calcium ions, and that's where the excitosis of the vesicles take place. Then once the vesicle is able to open into the synaptic cleft, the neurotransmitters that's within that vesicle opens up. It's in the synaptic cleft, and then it binds onto receptors that's at the subsynaptic membrane, which is the postsynaptic membrane. That means it's the second neuron. And so once the neurotransmitters are able to bind to this receptor, then the stimuli starts within the second neuron, which is where again sodium ion influx takes place, and then there's the stimuli that's transported within the second neuron. Now, the chemical synapse character, it's only one-way conduction. I think Tandis talked about it, the antidromic and the other one. And so in chemical synapse, there's only one-way conduction. You need the presence of a transmitter, and the mediator is proportional to the frequency of impulses. I'll come to that later, but what we mean is, Tandis talked about this as well, you need an impulse to generate the movement of this neurotransmitter to the next neuron. So the amount of neurotransmitters that are released in the synaptic cleft is determined by how many impulses like go through the neuron. So for example, if we have many stimuli from the first neuron, then you'll have more neurotransmitter which is released in the synaptic cleft. You'll have more receptors being activated by the postsynaptic membrane, and you'll have more stimuli with that passes on. So in the case, for example, your heart is beating. but you want it to beat faster, there'll be more of the catecholamines that's released. It's a very bad example if you guys still are not sure about what catecholamines functions are, but it'll make sense once you guys study. The synaptic delay, as you can see, is zero point 0.5 milliseconds, which is 10 times more than the synaptic delay of the electric synapse. And that means it takes longer. And it has high sensitivity to chemical substances and hypoxia. You also have another type of synapse called a mixed synapse. They don't ask you any questions about it. Just know what it is. It helps you for the final. Now, when I was talking to you about how the vesicle is transported, the neurotransmitter, right? It is what carries on the stimuli from one neuron to the other. These neurotransmitters, they can't just be moving freely along the neuron. It needs to be packed, and these packages are called the vesicles. You guys will learn this properly when you're studying for bio final. You just need to know like the very simple concept for physio, but for bio, you need to know this in very big detail. So what happens is the vesicle, I'm hoping you guys can see my cursor. The vesicle is like the circular like form, and it takes up the neurotransmitters, as you can see these small dots in the middle, and that's the uptake. Then you have the docking, which is where the vesicle is transported towards the membrane. There's some stimuli that causes for the vesicle to move towards the membrane, and this is calcium ion influx. If you guys watch Ninja Nerd, he explains this super well. And so once it's pulled towards the membrane, it anchors with the help of some proteins. And when this happens, the membranes of both the vesicle and the membrane fuse. And how this happens is with the T snare and V snare proteins. So T snare means target snare. Snare is just a type of protein that's present on the vesicle and the membrane. T snare is the target snare, so that means it's on the membrane, the presynaptic membrane. V snare is the vesicle snare. So that's the protein that's on the vesicle. And these two proteins, they bind together, and that's what causes the fusion to take place. And once it fuses, the neurotransmitters are able to move out into the synaptic cleft. Ignore this postsynaptic potentials part because I'm going to come to that later. Okay, so the next important thing is mediators. I know this looks like a lot of information, but I was too lazy to put this in many different slides. That's why it's all in one slide. The mediators, this is the neurotransmitters. Like I said, in the chemical synapses, you need the presence of neurotransmitters to move the stimuli from one neuron to another. You guys need to know the different types of neurotransmitters that's present. You have low molecular weight neurotransmitters, which is the most important, like acetylcholine, amino acid derivatives. If you guys study for some biochem, hopefully you will study. If you study, you guys will learn about this in very big detail. You have purines and you have substances of a peptide nature. So these are the three different types of neurotransmitters you can have. I'm going to go over the most important and the ones that they ask questions about. So first, acetylcholine. Acetylcholine is one of your most important neurotransmitters. If you ever see it, it's mostly going to be an excitatory neurotransmitter. That means it causes excitation. You can find it in these places. Guys, you need to memorize the places where you can find acetylcholine because this question comes a lot and everyone gets this question in the credit. It's found in the preganglionic and postganglionic. You guys can read this. I've listed it all down. These are the places where you can find acetylcholine. And acetylcholine is found in two types of receptors, muscarinic receptors and nicotinic receptors. Muscarinic receptors are found in the heart and the intestine mostly, and it's blocked by atropine. Nicotinic receptors are blocked by curare, and they're removed by acetylcholinesterase. This information, guys, you have to memorize. Like it's very easy to understand. Like there's this two different types of receptors, muscarinic, nicotinic. You're able to differentiate the two of them according to where they are in the body, and each of these receptors can be blocked by atropine or blocked by curare. And this question comes a lot in the credit. Why this is important, like to know these two different types of receptors, is if clinically, if you're going to explain it, for example, acetylcholine, like I said, it's mostly excitatory. So if someone is undergoing tachycardia, we're learning this in pathophys, I thought it was interesting. If someone is undergoing tachycardia, you're going to give them atropine because it's going to block these muscarinic receptors and the heart is not going to have as much excitation anymore. Then You don't just have acetylcholine. Another very important type of neurotransmitters is biogenic amine transmitters. And these are what I call catecholamines before. You have norepinephrine and epinephrine. These are the places where they're found. You need to memorize this. It's found in mostly the brain, but norepinephrine is also present within outside the brain. Even epinephrine, but just the spinal cord. Mm, you need to know that they are mostly they have a great effect on adrenergic receptors. So there's two different types of adrenergic receptors, alpha and beta. And under alpha you have alpha 1, alpha 2, alpha 3, and under beta you're going to have beta 1 and beta 2. In the lecture it explains this stuff. You need to understand that and you need to go through it. They ask questions on this. That's the only reason why. One of the other biggest questions that we were asked was the degradation. You can degrade norepinephrine and epinephrine by either oxidation or by methylation. You need to know the name of monoamino oxidase, catechol, ortho, COMT. You need to know these because these are two different ways where you can degrade norepinephrine, epinephrine. So when there's too much activity of the catecholamines, you have these two ways which will stop the stop their function overall. Then you have dopamine. You guys study this in ANAT. You see it in CNS. Then you have serotonin, which is in the brainstem. You also study this in ANAT. In the lecture, it was given this info. Just know the names like dopamine, serotonin, histamine, because they didn't ask too many questions about that. It's just you just need to know that they are neurotransmitters. It's mostly acetylcholine, NE, and E that you need to know about. Then you have amino acid transmitters. These amino acid transmitters are also important. You have glycine, glutamate, aspartate, and GABA. You guys study this in ANAT again, but you just need to know for your neuromyo credit that glycine is inhibitory, glutamate and aspartate are excitatory, and GABA is inhibitory. I don't know if you guys studied this during ANAT, but there was that whole cycle with all the basal ganglia, and you had these transmitters during that part. Okay, and this will come for your biochem. I just put it because I felt smart. You don't need to know the structures of this for physio. You just need to honestly just ignore this slide. You just have to know this information for the neurotransmitters. Now, guys, any questions so far about this? No? Okay, perfect. If you guys do end up having questions, you can always like type on the chat box. Okay, so then we're going to talk about the relationships, the relations between the different neurons. So there's a very simple part of neurophysio, like it was the most easiest part. So we have convergence and divergence. Convergence is where many different neurons, like presynaptic, you have many different presynaptic neurons giving stimuli to one postsynaptic neuron. You just have to know that. Convergence is many neurons to one neuron, and divergence is the opposite, where one neuron has many axons that is able to give stimuli to many postsynaptic neurons. So convergence was from many to one, while divergence was from one to many. It's just the stimuli passing on. Then we need to know about summation. So what I mean by summation is there are some instances in the body where there needs to be more stimuli present. For example, when you're in fight or flight, when you're in a dangerous situation, you'll be more alarmed. Your heart will be beating more faster. So you need stimuli that actually stimulates this increased activity. So to see this increased stimuli, we have something called summation. Summation as in summation. And you have two different types of summation. You have temporal summation and you have spatial summation. Temporal summation means that within a certain frame of time, there's going to be more vesicles and more neurotransmitters that's sent to the postsynaptic membrane. So this will give you like an increased stimuli overall on the postsynaptic membrane because there is more stimuli present. And why it's called temporal is, so within a certain frame of time, in a certain frame of time, there's more transmission taking place. Spatial, however, is when there's more presynaptic membranes. Temporal was one, it's just there's more release from that one. But spatial is there's many presynaptic membranes that's sending the same amount of neurotransmitters, which overall on your postsynaptic is going to have the same effect. You're going to have more stimuli being stimulated. Now, what this does is it causes a postsynaptic potential, which it can be an EPSP or an IPSP. What's an EPSP? It's an excitatory postsynaptic potential. So what that means is when the neurotransmitters are excitatory, that means they want you to, for example, heartbeat. They want to increase your heart contractions. EPSP is going to be more increased excitatory. So it's going to be even more tachycardia and it's going to cause a greater action potential. While IPSP is inhibitory and it will cause less. So it will cause bradycardia instead. I know this sounds like a lot of words, but it'll make sense once you guys go through it on your own. Then we talk about occlusion. Occlusion is a theory in which, for example, now I was telling you guys about divergence and convergence, right? In the case of convergence, right, you'll have, for example, let's give a number. You'll have two neurotransmitters sent from the first, two neurotransmitters sent from the second, two neurotransmitters sent from the third, and two neurotransmitters sent from the fourth. So overall, you have 2 plus 2 plus 2 plus 2, which is 8. You're supposed to see the activity of 8 neurotransmitters in the postsynaptic membrane, but you don't see that. There's only 6 neurotransmitters. And that's what occlusion describes. It means that even though there's a certain power that's sent from the presynaptic membranes, the postsynaptic can only take a certain amount. This could be because of the lack of receptors. Like there are neurotransmitters in the synaptic cleft, but already all the receptors already are filled. So the other neurotransmitters just have to like vibe, you know? So that's what occlusion is. Then you have post-tetanic potentiation. Post-tetanic potentiation is where if impulses are conducted through a synapse, then the rest is given to the synapse. Then again, impulses are conducted. It's just post-tetanic potentiation is when there is increased facilitation of stimuli. It just keeps building up. Tandis talked about the absolute refractive period and the relative refractory period. In the absolute refractory period, there's no excitation that can take place. But in the relative refractory period, there is excitation taking place. So if there is very quick stimuli that's continuously sent, you'll have a graph that looks like, okay, there's one upward excitation, another additional excitation that comes on top of it because it meets the requirement of being in that relative refractory period. And this is it's quite complicated when I'm explaining it. I think once you see the graph and once you watch Ninja Nerd, he'll explain better because he draws it on the graph. It's just the whole point is that you're gonna get a continuous increase of excitation, and I hope you guys know what tetanus is, because this is what causes tetanus in your heart. If you have so much excitation like this, that's what causes tetanus. Then you have the inhibition. So we talked about the excitation. We did talk about inhibition a bit, but you have postsynaptic inhibition and presynaptic inhibition. So postsynaptic is where it's after the synaptic cleft is present. So you're inhibiting the stimuli once it's already reached the second neuron. But the presynaptic inhibition is if the stimuli does not reach the threshold. For a stimuli to pass from one neuron to the next, it needs to meet the threshold, which is what Tandis talked about. If it does not meet this threshold, the stimuli does not pass through. And if it does not pass through, there's no stimuli at all. So there's presynaptic and that's postsynaptic. Okay, I'm gonna briefly start up the muscle physiology. You guys have done this in histo. It's super, super easy. The only difference with the muscle physiology is you go a bit into depth of what happens between the actin, the myosin, but especially smooth muscle was a bit different. But it's again, they mostly just ask you basics in the credit. So you need to know that the muscle types, you guys have learned this before: skeletal muscle, smooth muscle, myocardium. I hope you remember that from histo. Skeletal muscle has syncytium. I hope you all remember. Does anyone remember what syncytium is?
I think
Everyone's
no
so
one.
collaborative today.
Yeah, no one remembers. I think just explain.
Syncytium is
I
where
think I think group of nuclei.
Yeah, it is the many nuclei. Thank you, Alaa. So, since it's here, you need to know the several proteins in the skeletal muscle. You have the contractile proteins, you have regulatory proteins, you have the accessory proteins. Once you guys watch the playlists, he explains all the functions, and you guys know what all of this is. You've heard of it before. You need to know about the connective tissue of the skeletal muscle. The surrounding is a surrounding of one of specific reticular fibers was endomysium. Perimysium was a group like between bundles. It separated the bundles. Epimysium was on the surface of the muscle. Then you need to know that the innervation of a skeletal muscle, this is really important because I remember getting questions about this. One alpha motor neuron is able to innervate a specific number of muscle fibers, and that whole part where like one alpha motor neuron and innervating a few muscle fibers, this was called a motor unit. This is also really important in CNS. In the motor end plate, that means the connection between the nerve and the muscle, you have the neurotransmitter acetylcholine. So you get to apply what you learned with the neurotransmitters here again. And its receptors are nicotinic, not muscarinic, but nicotinic. And if you guys remember, curare blocks the receptors. So that's how you're supposed to apply what you're learning in neurophysiology with myology. Now, there's many different changes that happen during muscle contraction. There's structural, chemical, thermic, mechanical, and electrical. I'm just going to go over the structural, and Ella is going to take it after that. Um, so the structural, you guys already know the structure. You learned it in histo, you learned it in anapt. You have the A band, you have the I band, you have the H zone, you have the Z disc. So everyone knows that the A band is with the myosin, while the I band has the actin, and the H zone is just alone the myosin part of, like, without the actin overlapping. So it's just the part of the myosin, and the Z disc is the connection between the actin filaments, and I band is just the actin, while A band is just the whole myosin with the overlap of the actin. We've already learned this many times before. And you also need to know about the terminal cisternae and the T-tubule, the triad. Just go over what you guys had learned in histo and you'll be good. And yeah, that's all for me. Ella, do you
Katie,
want me to stop sharing?
do you want to keep sharing screen? And like, you can just
Yeah,
move
I
when I'm
don't
done with
mind.
this one. Okay,
I
perfect.
don't mind. Give me a second.
All
I'm just
right.
gonna unmute.
Okay.
Give me
All
one
right.
minute.
So for the last part of this, I'll try to make it quick. It's going to be mainly about myology, so muscles, obviously. And it's just going to be adding on to what Tandis and Katie already explained, but like in more details and also more related to muscles. So let's start with muscle twitch. So what is a muscle twitch? By the way, this exact part I remember they asked about it a lot during the credits, so focus on it. So it's basically a single brief contraction and relaxation of a muscle fiber after a single stimulus. All right, so I think simple, easy. So contraction, relaxation, following a stimulus, which is what usually happens, but in here it's more specific. So what are the phases of this? So we have three phases: latent phase, the contraction phase, and the relaxation phase. All right. So the latent phase, obviously from the name, it's the time between the stimulation and the onset of contraction. So the time between when there's a stimulus and when the contraction actually starts. Then contraction phase, obviously contraction. So muscle fiber shortens as actin, myosin interact. I'll explain this more in following slides. And then the relaxation phase. So that's when the muscle fiber goes back to its resting and normal length. Now, what are some factors that can influence twitches? So the stimulus strength and frequency, the muscle length before contraction, the temperature, pH, and ion concentrations. I'll explain this specific point in later slides. An example of the factors would be that increasing frequency would result in summation, which Katie described briefly, and I will also describe. But Katie, you can go to the next slide. And by the way, guys, if you have any questions, you can write on the meeting chat or you can raise your hand because I can see if you guys type anything. So what is summation? So summation is increased tension because of successive stimuli before relaxation completes. So basically, in simple terms, what does this mean? So as we said, we usually have stimulus, contraction, relaxation. And then if there's another stimulus, the cycle will repeat. However, what we're saying here is that there was stimulus, contraction, stimulus. So we didn't reach the relaxation phase yet. And so we call this summation. And I believe there are graphs on this in your physiology PDF. So see them and know them because they might ask about them. Now, tetanus, Katie described, but I'll go into two different categories. So incomplete tetanus is when there's partial relaxation between the stimuli. Complete tetanus is when there's sustained contraction with no relaxation at all whatsoever. Okay, Katie, you can go. All right. So synaptic transmission, Katie and Tandis already described this as well, but I'll read again just as a quick recap in simpler terms. So neurons, obviously, they're not into direct contact, but they send signals across synapses or in small gap, synapses which are small gaps, and the signals are transmitted through neurotransmitters. The signals are either excitatory or inhibitory, which is self-explanatory, I think. And then post-tetanic potentiation. It means that there's enhanced muscle response after repetitive stimulation. And this is why we see sportsmen warming up before actually exercising. It's to enhance the muscle response by stimulating it before they actually start the exercise, whatever it is. And what is the mechanism of that? It's by increasing calcium availability in the sarcoplasm by using the muscle, causing it to contract before the actual exercise. Okay, Katie, you can move. All right. This slide has already been described, so I won't explain it again. You can move. This as well. You can move. All right. This okay. So now we spoke about, since we said that I'm going to focus on myology, so we know that the major role for a major key player in contraction is calcium. So calcium channels are critical for muscle contraction and neurotransmitter release, because without those things, we won't have muscle contraction at all. So DHP, which is dihydropyridines, they can block specific calcium channels and cause relaxation of muscles. Because as we said, if there's no calcium, there's no contraction, right? So now we have two types of muscle fibers, right? So we have type 1 and type 2, obviously. One is slow twitch and 2 is fast twitch. So what is slow twitch? So it's oxidative, so it uses oxygen for long duration activities, like running a marathon. However, fast twitch generates quick, powerful movements, but it fatigues quicker. Now, in fast twitch, there are, again, two types. So type 2A and type 2B. So right now we said there's type 1 and type 2, and then type 2, there's A and B. Now, A is oxidative and B is glycolytic. Katie, you can move. Okay, so smooth muscle physiology. So smooth muscle, again, we're going to classify. So it's either multi-unit or single unit. So multi-unit is controlled independently, like in the eyes or in the blood vessels, and it allows fine adjustments. And single unit muscles, they work together, so not independently. They work together by being interconnected through gap junctions. And we see this in the GIT because there we have peristalsis. So we need rhythmic contractions. We need them to be in coordination with each other. Now, how are smooth muscles different than striated muscles? So obviously there are no striations. That's an obvious one. And they also contract more slowly, but they can hold the contractions for a longer period of time. Katie? All right. Now, cardiac muscle is striated. It's not smooth, obviously. And they have special connections, which are intercalated discs, which I remember were heavily focused on during histology. And these discs, they synchronize contractions with each other. Their calcium channels stay open for a longer period of time. So that creates a plateau in their electrical activity. And that gives us steady and strong heartbeat. Now, sarcomeres were briefly shown, I think, in the diagram that Katie had. So let's speak about them more. So sarcomeres are the functional unit of muscle contraction, and you can find them in striated muscle, so cardiac and skeletal muscle. Now, what are the components of these sarcomeres? By the way, the diagram that Katie showed is extremely important, so guys, make sure to know it. So anyway, back to the sarcomeres. So the components are actin and myosin, and also some regulatory proteins like tropomyosin, troponin, and Z-lines. I'm sure all of us already know these things from histology in specific, but make sure to cover these things again, guys. Okay, Katie, you can move. Okay, so let's speak about calcium again. So what does calcium do exactly in muscle contraction? How does it start it? So firstly, what happens is we're gonna have some sort of stimulus, and then we're gonna have action potential traveling down motor neurons. And then acetylcholine is gonna be released into the synaptic cleft, which is the gap that we spoke about. And then that causes the muscle membrane, which is the sarcolemma, to depolarize. And then that depolarization is going to cause sarcoplasmic reticulum to release calcium into the cytoplasm of the cells. And then calcium will bind to troponin C. And then that binding will cause a change in the troponin complex. And then this change will cause the tropomyosin to move. And then that will uncover a site on actin. And myosin can bind on that uncovered site. Now, I know this might seem a bit confusing, but try to know it as like order of events. So it happens like 1, 2, 3. So that will make it less confusing for you guys. Okay, Katie, you can. All right. So how does the contraction actually happen? What are the cross bridges being formed? So as we just said, a certain site has been uncovered on actin. So now myosin is gonna the myosin head in specific is gonna bind to those exposed sites, and then the head is gonna release ADP and inorganic phosphate, and then it's gonna pivot, so it's gonna move, and when it moves, it's gonna pull the actin filament toward the center of the sarcomere, and that's gonna shorten it because they're gonna slide over each other. And then when a new ATP molecule binds to the myosin head, it's going to detach from the actin, which obviously will stop the contraction. And ATP is going to be hydrolyzed to ADP and inorganic phosphate. And then that can move the myosin head back into its high energy state. And then as long as we have calcium and ATP, the cycle can continue. Katie, you can move. Now, how about muscle relaxation? How does that happen? So the calcium is actively pumped back into the sarcoplasmic reticulum by calcium ATPase pump. Because if we don't have calcium, then we're not going to have contraction. This will decrease the levels, obviously, and it will detach the calcium from the troponin. And so the entire cycle we just spoke about won't happen. And tropomyosin obviously covers myosin binding sites in actin, which we've already spoken about. And during relaxation, the elastic components and the muscles are gonna help the sarcomere have its original length. So it's not gonna be shortened, so it's not gonna contract. Okay, if you know. Okay, this is basically what we just explained, but in a table. If you guys want to read it later to, I don't know, simplify the information. But yeah, like I am not going to go into it again now. I can move. Okay, so energy sources for muscle contraction. I think we already know this, but just a quick recap. So ATP, phosphocreatine, glycogen and glucose, and fatty acids. All of them can be used as energy sources for muscle contraction because obviously muscle contraction is an active process and it needs energy. Okay, Katie, you can move. All right, now the environmental factors that I said I would go into later, I'm going into them now. So pH, temperature, and hormones. So for pH, the acidity can influence how excitable nerves are. And if the blood is too acidic, nerves will work less efficiently. So the more acidic, the less signal transmission there will be. Now, about temperature, cold temperatures can make nerves more excitable. That's why when you're cold, you shiver. As for hormones, substances like adrenaline can reduce the excitability of nerves in some situations, helping you stay calm or steady during stress. So we know about sympathetic nervous system being fight or flight and parasympathetic being rest and digest. So it's relevant to that. All right, now for the ANS, I'm going to briefly describe it. but there's way more detail in your lecture, so please go into detail regarding it. So ANS is the autonomic nervous system, obviously, and it controls things that we don't consciously do, like heartbeat, digestion, breathing. Obviously, you're not going to think before you start digesting or before you breathe or before your heart beats. It just happens. Now, adapt to things that happen around you, so either externally or internally. And as for the effectors, the ANS acts on smooth muscles, like in blood vessels, cardiac muscles, and glands. Okay. And these result in some sort of effect. Now, as for reflexes, reflexes are rapid automatic responses to certain stimuli. And we have reflexes to either protect the body or to just maintain homeostasis. Now, we need to know that reflexes are obviously involuntary and they immediately happen, and they don't involve higher brain centers initially. So what is an example of a reflex? If you touch something hot, you remove your hand without thinking. So that's a reflex. And this diagram that I put here is in your presentation. So make sure to understand it because it explains the general concept of reflexes. Okay. You know? All right. So what are the components of a reflex arc? So firstly, what is a reflex arc? It's the neural pathway in which a reflex travels. So we have 5 key components. The receptor, which detects the stimulus. The sensory neuron, it takes the information and the impulse back to the CNS. The integration center, it processes the information that it just received. And that happens in the spinal cord or the brainstem. And the motor neuron sends the response signal to the effector, and the effector performs the action. And the effector is usually a muscle or gland, as we explained previously. Katie, I think it's in the end slides of the presentation that they have.
This
The
is
PowerPoint.
oh
Yeah, go back.
no, because that stuff is in CNS.
Okay, so it's not covered in this test.
No, like the thing is we studied for it, but even after I think we restudied again for CNS.
Ah, okay. All right, perfect.
So this part, thank you, Alaa, it was really good. I think just this last part is just CNS. So we'll do it over
Okay,
again
perfect.
when we're ready for them.
Okay. That's it for me. If you guys have any questions, you can ask us in the chat or you can unmute and speak. No?
I think we can ask them some questions to
Okay,
see if
sure.
they're actually listening, because you know they were so like interactive.
Okay.
Okay. All right.
Okay, let's see.
We have 2 minutes, yeah, to ask questions.
Okay, yeah. Who knows the molecule that binds to split the connection between actin and myosin? It's an easy one.
Yeah, I just said that.
That's
Yeah.
so easy.
No one knows that?
Calcium?
I said calcium. No.
No.
Yeah. Yeah, it's calcium. But
Huh?
it is calcium.
It is?
That all binds.
It's calcium and troponin.
I think Ella, you and me are thinking of the other part of unbinding.
Okay, that works
Guys,
as
the
well.
unbind at the beginning, yes, it's troponin and calcium, but then to unbind after contraction, it's ATP.
ATP, yeah.
Yeah, that's true. I didn't say like before contraction or after contraction.
Yeah. So this is how they'll get you in the questions. If they just say like unbind, you think of all of this.
Yeah.
Okay, but thank you guys for attending.